Now, an update on HIV/AIDS. Let me review a review or two reviews of biological factors for the HIV pandemic. Other people also thought that it has to do with biology, not with social behavioral factors or political factors, economic factors in Africa. Rupert Call especially also wrote a review with others that it has something to do with blacks. They call it race even. I prefer ethnic group or people that live somewhere because it's anthropologically and philosophically a less controversial term. Now, what does Rupert Call think? Let me review that. The first factor that they mention for the spread is the clade of the virus. A clade in biology is a family of something. And the family term then is based on a common origin, on a common development or movement of a biological entity in space and time. That's, I think, what a clade is. And so the HIV virus, as other biological entities, also has clades, different clades, and Clade C of HIV is mainly present in Africa and in India, and is not so common in other places of the world. And so maybe it's the clade of the virus that's responsible for that enormous spread in sub-Saharan Africa. But the authors here describe that themselves as an eminent factor because blacks are also the main victims in places where Clade C of the virus is not so common, like in North America. I might also add, they don't mention that, I think, in India with Clade C, where Clade C of the HIV virus is pretty common, the total numbers of HIV positives are high, but the prevalence is rather low relative to the population. The total numbers are high, but the prevalence, the relative prevalence is almost nothing compared to sub-Saharan Africa. The second factor that they discuss is genetic factors. Now, the main factor that they mention here is the CCR5 receptor mutation. The CCR5 receptor is needed for the HIV virus to enter the CD4 positive cells. They need the CD4 receptor and the CCR5 receptor, and there is a mutation, the CCR5 delta 32 mutation, and when you have that mutation, the receptor is broken, can't use it, or HIV can't use it, and it doesn't get in the cell that easily. But unless some other genetic factor is found that is really very common in non-blacks and rather uncommon in blacks, this mutation cannot be an eminent factor, simply because, I mentioned this already in the other videos, simply because non-blacks reach about the same, well, maybe a little lower, but non-blacks reach about the same infection rates simply by being anoreceptive MSM or IV drug users, simply by showing a certain risk behavior. So race per se or ethnic groups, ethnicity per se does not protect, cannot be a major factor. And my main argument against such genetic immunological factors is again the finding that old sex workers did not catch HIV that or as much as young sex workers. The immunology, the genetic makeup cannot change within 10 to 20 years. That would be my own main or my favorite refutation of immunological factors or genetic factors in Africa being responsible. Well, it is genetic factors, but only insofar as it is the genes that contribute to the genital organ variant, which would now be my favorite hypothesis. The hypothesis that pleases me, as someone put it, but it doesn't. I don't embrace it because it pleases me, but because it's simply plausible. Now, they also mention another genetic factor, which is the CCR2 receptor. It's a chemokine receptor, and if there is a mutation in that one, you also have a protection, but there the results are in a way the other way around. If that receptor mutation were responsible, then you would actually expect that there are fewer infections in sub-Saharan Africa than in Europe, because it's more common in sub-Saharan African people that in that mutation than in Europeans. Then they also mention co-infections. This is the third factor, or the third group that they mention category. The co-infections in Sub-Saharan Africa. They are the biological factors in Sub-Saharan Africa. And they say the viral load is increased with tuberculosis and malaria, which are present in very high numbers in Sub-Saharan Africa. And that could indeed be an interesting possibility. The viral load of HIV is increased with other infections, and so the infection is more likely. If you have more viruses present in the bodily fluids, then transmission is easier. So that's an interesting possibility, I admit. Or also the point that STIs increase the number of activated CD4 positive cells, especially in the anogenital sites. That's also interesting as a hypothesis. Then transmission is easier, is more likely. There is also a possibility that CD4 positive cells could in the anogenital sites also be elevated in non-sexual infections like malaria or helminths. But this is not too clear, the authors say. But in my opinion, they can save, or people can save the work of finding this out, because again, that doesn't explain, it wouldn't explain the preference for blacks in other areas of the world where such infections are rare, helminths or malaria. You don't find such a high prevalence of them in North America, where blacks still are one of the main victim groups. Yes, tuberculosis and malaria, they increase the viral load, but that doesn't explain that blackotropy in other parts of the world. Also, yes, the other STIs, not tuberculosis and malaria, they increase the number of activated CD4 positive cells. But now, again, where do these STIs come from? I also like their smartness of considering whether the non-HIV STI prevalence is actually also higher in Sub-Saharan Africa and in blacks in general. Because, after all, if STIs are responsible, they should be more common in the first place. And then they found studies showing that yes, they are. They are more common. There are more STIs in blacks in general, and also in Sub-Saharan Africa. But still, you would have to ask one step or question further. That result with a higher number of CD4 positive cells in the anogenital sites, when you have other STIs, that point or finding is of no use if that STI thing itself is not explained. Especially, why are there higher numbers? Yes? It's a good thing that they address the question at all. Are there more STIs in the first place? But then one question further would have to be asked: why that? Now, as for the causes for this higher number of STIs, there is no answer. The authors here in that review state that the reasons for this are poorly understood. For example, why bacterial vaginosis in blacks has a higher prevalence. Now, I can tell you what causes bacterial vaginosis in blacks. That bacterial vaginosis is that they are simply sore because they're tighter at the introitus, or maybe along the whole length of the vagina. And these little mucosa cracks that result from this tightness provide a better nest for, or start an immunological fight against, the Gardnerella vaginalis bacterium that almost all women have in their vagina anyway. This might also be a reason why Rappert Call found elevated CD4 positive cells vaginally in otherwise healthy sub-Saharan women in another study of his. There is another review on biological factors in HIV transmission, and that was done by Michael Marmer in 2006. And what they discuss over and above the factors that have been discussed by Rappert Call here is that they also mention cytokines, interleukin 4 variants, but these seem to be playing a role mainly in Japan, I think I remember, in Japanese people, and also for the progression of the disease, and not so much for the infection itself, for the transmission. And also the results have been controversial and could not have been replicated in homosexual groups, for example. So the results are not clear. They also mention HLA variations, human leukocyte antigen variations, that may also play a role, but here also the matters are somewhat complex, and these HLA variations are not mentioned in the 2011 review. So I assume that they don't play that much of a role. And also, HLA variations are also associated with autoimmune skin conditions, and so this is where there could be a link to my theory of genital organ variations and skin variations in the genital organs. But now my initial research on this was not very fruitful, as the HLA variations that are associated with scleroderma and such seem to be somewhat paradoxically, from me or my hypothesis, they seem to be associated with a protection against HIV transmission. So this part does not really fit, but maybe the role of HLA variations is not at all that important. Again, because it was not mentioned in 2011 paper, and because it's very intricate. These matters are very intricate. So this is my take, or this is my judgment on biological variations. In summary, it doesn't impress me very much as an alternative hypothesis to mine. I think my explanation is more plausible, explains many more things than do these biological factors.